Hello guys, today we are going to discuss molecular biology day 1 in Virul Weerakoon's revision. First I am going to go through the main points that we have to go through. So the CT 117 basic principles of molecular biology. Under basic principles of molecular biology we are discussing about structure of DNA molecule and then DNA RNA conversions. Then we discuss about instances of conversions, instances of where DNA RNA conversions happen. And fourth, we talk about DNA and how some enzymes are helping it to replication and other related processes. As the fifth point, we are looking into the structural arrangement of DNA, and then we are looking into DNA packing, how DNA is packed inside cells. Then finally, we discuss some conversion questions. Right. Basic principles of molecular biology. First one, structure of DNA molecule. So when we take a DNA molecule, we can see that it is made up from two strands. We call it double stranded, right? If we take a DNA molecule like this and then we unwind it, we can get two strands. Hence it is a double stranded molecule, and it is also double helical. Double helical meaning the DNA molecule is made up of two helix. So this is a single helix, right? And this is another helix, so it is made up of double helix. We call it double helical. And also two anti-parallel strands. We just discussed the DNA molecules is made up of two strands, double strand, but now we are discussing about how they are arranged, right? They are two anti-parallel. Because they sit next to each other, we can call them parallel, but not just any parallel, anti-parallel, right? If a strand runs from 3' to 5', the other strand runs from 5' to 3'. So here's an example explaining this anti-parallel. So here a kid is standing usually, and here he is standing from his toe to head, here head to toe. So these two kids are parallel, but they are not just any parallel, they are anti-parallel. So this explains it. So DNA is similar, and DNA also has the ability of directing, right? Directing means if somebody gave a DNA molecule to you and they broke it down, and so they gave you a single strand, right? We have DNA molecule, somebody broke it down and gave you a single strand, right? Let's take X, right? Somebody gave you X strand, and now they ask you what is the directing of the other strand, or in this case X dash strand. So if you know that if you know the directing of X strand is from 3' to 5', you definitely know the other one should be, other one must be other one must have the arrangement running from 5' to 3', and you can just decide it without looking at it, right? And that we call the ability of directing, right? We can predict the direction of X dash strand because of the directing ability of DNA. If somebody give you X, you can find X dash without even looking at it, right? That is the ability of directing. And then we have to know eukaryotic DNA and prokaryotic DNA. So the difference between these two is that the eukaryotic DNA is linear, prokaryotic is circular or cyclic. And then we have to look at complement base pairing in DNA. So there are 4 bases: adenine, thymine, cytosine, guanine. And adenine always pairs with thymine, creating 2 hydrogen bonds. Guanine always pairs with cytosine, creating 3 hydrogen bonds. This is in DNA. If we move into complement base pairing in RNA, instead of thymine, here adenine pairs with uracil, creating 2 hydrogen bonds. So in DNA it is adenine and thymine, right? DNA RNA conversion. So we have 1, 2, 3, 4, 5 points under this DNA RNA conversion topic. So first we have to know that the whole idea behind this molecular biology subject is that we have genetic material on the nucleus of a cell, and using this genetic material or DNA, we have to code for proteins. We have to create proteins. This is the base of this lesson, right? When converting DNA into proteins, you have to also follow through the intermediary step of creating RNA and other substances. So the 5th point is the explaining the whole base of this lesson. And if we move on to conversions, we have DNA to DNA conversion, and we call the process replication, and there are enzymes involved in this process. And DNA to RNA conversion, the resultant RNA is called mRNA. The process is called transcription. If we convert DNA into RNA, the process is called transcription, and also enzymes are involved. Then we are doing the reverse of that. Here we convert DNA to RNA. Now we are converting RNA to DNA, right? the resulting DNA is called copy DNA or cDNA. And obviously the name for this process is called reverse transcription, because converting DNA to RNA is called transcription. Hence this is the opposite or reverse process of that. No, converting RNA to DNA, so it is obvious that the process name is reverse transcription. And here also enzymes are involved. The enzyme involved is known as reverse transcriptase. And then we move into RNA to RNA conversions. We have mRNA here and we have tRNA here. So this take place, RNA to RNA conversion take place in ribosome subunits. And so the thing about mRNA, right? mRNA is that it transports its genetic material as codons, right? Codons are base triplets, right? And tRNA transports its genetic materials using anticodons. It is also base triplets. Both codon and anticodon are made up from 3 bases, like you might have heard AUG codon, like that. So these are made up of 3 bases. If it is mRNA, we call it codon. If it is tRNA, we call it anticodon. And tRNA is used to create amino acid right here. So tRNA, we have anticodons. The conversion of tRNA and utilizing tRNA to create amino acids, the process is called translation, right? Process is called translation, and that is the content of this slide. Then we move on to instances of conversion, right? Instances of conversion where these conversions happen. It can happen either on living system or non-living system. Non-living system we can also call artificial environment. Examples for living system is cell. And the conversion which happened in living systems, so inside a cell, we call it in vivo. And conversion that happen in non-living systems or artificial environment, such as a test tube, we call it in vitro. Here in vivo, here in vitro. And DNA and enzymes, the relation. So we have to do some stuff into DNA to do replication and all those things. And DNA utilize the help from enzymes to get it done, right? So first we are looking at the enzymes used in different types of processes, right? First we have DNA molecule right here. There are instances where we have to remove the helix, right, or unwind. Here enzymes are involved, and then after we unwind it, we have to break the bonds. Unwinding, and now we have to break the bond. Here also enzymes are involved, and sometimes we need to cut this large DNA into smaller parts. We call it cutting DNA. Enzymes are involved here too. And then usually when we break the bonds of the 2 strands, usually it tries to go back to its formal structure, which is the formal DNA molecule, meaning they try to wind back into the original structure. We have to prevent that. And enzymes are also involved in preventing the double strands which bonds are broken, preventing it into going back to its normal structure. We also use some enzymes. And then we know that DNA molecules are formed from polymerization of nitrogenous bases, right? For polymerization we also need enzymes. And sometimes we need to bind fragments. So we have smaller parts of DNA, and we need to bind them together and convert it into a larger DNA. So enzymes are involved also here. And you know this process, right? DNA and RNA. So in this process, DNA and RNAs are sometimes mixed up like this. So we have DNA segment here, now we have RNA segment and DNA. So we cannot leave it like that. We have to replace the DNA RNA hybrid with pure DNA into a pure DNA molecule, and we use the help of enzymes to get it done. So E1, E2, E3 does not mean the actual names of enzymes. The E1 here in means that some kind of enzyme is used here, and not the actual enzyme name, right? Structural arrangement of DNA. So in this lesson, I told you before that this lesson is based on RNA, DNA, and proteins and interaction between them. So structural arrangement in DNA, you have to know that there are 2 types, which is linear and cyclic or circular. Linear means there is a starting and ending point. In proteins, you have to know that there are histone proteins and the rest of the proteins. So there are 2 types, which can either histone proteins and the other proteins. DNA packing, right? DNA packing into cells. In eukaryotes, the DNA is stored within a nucleus, bounded by a bounded by the nucleus, and in prokaryotes, DNA is not covered by a nucleus. It is spread throughout the cytoplasm of the prokaryotic cell, and the region which is which genetic material or DNA is spread is called nucleoid. Finally, we move on to conversions. In conversions, the first question is about question 1. Convert this DNA sequence into the complementary DNA sequence. So adenine pairs with thymine, guanine with cytosine, and again thymine with adenine, cytosine with guanine, cytosine with guanine, guanine with cytosine, adenine with thymine, guanine with cytosine. So this is a single strand. This is the complementary of the other strand. And question 2 is asking, so here's a single strand, asking us to write the other strand, also give the number of hydrogen bonds made. For thymine, we have adenine, thymine, adenine, adenine, thymine, cytosine, guanine, guanine, cytosine, thymine, adenine, adenine, thymine, guanine, cytosine, cytosine, guanine, guanine, cytosine. So adenine, thymine makes 2 bonds, cytosine, guanine makes 3 bonds. When we calculate all of these bonds, we have 25 bonds here. And the question 4 is asking us, so we have given a DNA strand. The question is the mRNA. Question is asking the mRNA of complementary DNA molecule, complementary DNA strand. So it is not asking just the mRNA of the DNA molecule, it is asking for the DNA of complementary DNAs, right? I mean, it is a little bit confusing, but you will understand it hopefully when I explain, right? You have a DNA molecule like this, and first we have to convert DNA into mRNA. So when converting DNA into mRNA, we have to know that thymine is not available in mRNA. The replacement is uracil. So uh, if some place seems like there should be thymine and it is mRNA, it should be uracil, right? Here adenine pairs with uracil, not thymine, because this is mRNA. Guanine is cytosine, thymine is the adenine. It is okay because thymine is on the DNA molecule, and complementary adenine is available in mRNA. Only uracil, only thymine is not available in mRNA, right? Thymine can be available in DNA. Guanine, cytosine, and adenine, again it's not thymine, it is uracil. Thymine, adenine, guanine, cytosine, cytosine, guanine, adenine, then uracil, thymine, adenine. Then using this mRNA, we make the copy DNA or DNA dash. The DNA dash is uracil, uh, uracil, uracil, thymine, cytosine, cytosine, adenine, adenine, guanine, guanine, cytosine, cytosine, uracil, thymine. So only different is this: the uracil converts into thymine, right? It is not complementary; it is just the same bases. The difference is uracil is converted into thymine, right? Copy DNA. And from this copy DNA, we are asked what is the mRNA of that copy DNA, mRNA dash. So the question is, you are giving this DNA, you have to ask, you are asking, the question is asking what is the mRNA dash, or the mRNA made from the copy DNA. So the mRNA of copy DNA is thymine, gives us pairing with adenine, cytosine with guanine, adenine with uracil, guanine with cytosine, cytosine, guanine, thymine, adenine, adenine with not thymine, it is uracil in mRNA, cytosine, guanine, guanine, cytosine, thymine, adenine, adenine, uracil. Right. Now you see the only difference between this DNA and mRNA dash is that thymine is replaced with uracil, right? So that is the only difference. If you can remember this, and if a certain kind of question is asked, you just don't need to spend time this writing this. You just know how it is done. You just have to replace thymine with uracil, right? We also have a this also explain the same thing in a simpler way. I hope you got an idea, and thank you.